So the point of the Emeriti lecture is to select, we have 2 of these a year. We select, the group selects a speaker from the Emeriti, and it illustrates to the campus and the community that Emeriti continue to do research and remain active even after retirement. And tonight's speaker is, as you will see, is a fine example of that. This is sponsored by the Emeriti Group, but the funding for it comes from the Chancellor's office. And I'd like to thank the Chancellor for supporting this, and also the reception that follows. I'd also like to thank Kristen Palma from the Special Events group who helped to organize this, arrange the publicity, and also the reception afterwards, and arranged to have it videotaped, so it will be recorded for posterity in the library. And some of the previous lectures have actually found their way onto YouTube or on TV afterwards. So I invite you to stay after the lecture for the reception and to talk to people. Our speaker tonight is research professor David Deamer. He's in the Department of Chemistry and Biochemistry and Biomolecular Engineering, which is a new department that's located in the School of Engineering. He actually helped start that and served as acting chair of the department for about 5 years. Going back, his undergraduate degree was in chemistry at Duke University in Durham, North Carolina, and then he went on to a PhD in physiological chemistry from Ohio State University in the School of Medicine. So he started out chemically and got more into the biological domain as time went on. Following that, he did a postdoctoral at Berkeley, and just at that time, the University of California was in great turmoil. Clark Kerr had just been fired by Governor Reagan, and he was looking for a job. And he found one at Davis University in the Department of Zoology. There he started as assistant professor and rose to the rank of full professor. He chaired the department for a while. And in 1994, he moved his research group to UCSC. He took early retirement, the 3rd BARUP, and became a research professor here, which basically meant the university didn't pay him. He just did research and continued here. So as I said, he was acting chair of the biomolecular engineering department, and he has continued to have students and postdocs. Since he moved here, he's mentored 9 graduate students and 9 postdocs, as I counted them. And he's looking back in his career, his excellence that we see today was recognized very early in his career, because he was selected by the Westinghouse Science Talent Search as in 1957. So their search engine was a good one in picking out a winner. He's won many awards, including a Guggenheim, which he spent at the Australian National University. And he has, of course, lectured at universities all over the world. He's on currently three editorial boards: the Journal of Bioenergetics and Biomembranes, the Journal of Astrobiology, and the Origins of Life and Evolution of the Biosphere journal. So this gives you a flavor of things that he's interested in. He has two interesting patents. He actually has four, but I'll just pick out two. One is called molecular barcodes, and another is single nanopore channels. And the latter relates to one of his two main research areas, which is concerned with DNA transport across through a nanopore membrane, in a membrane, and the ability to develop an instrument that might be able to analyze DNA rapidly as a single molecule and even perhaps sequence it. So this, when perfected, might make sequencing your genes a couple minute or 10-minute operation and quite inexpensive. The second research area that he has is the one he's going to talk about tonight. Basically how cellular life started on Earth nearly 4 billion years ago. And I'm not going to discuss that in the introduction because he's going to be talking about it, but only to say that he's in the process of writing a book on the origin of life that will be published by the University of California Press. And some of the scenes, some of the pictures that you're going to see are a preview of that book. They're taken from the book, he tells me. You might wonder who funds research on the origins of life. And he is supported by the NASA astrobiology program at NASA Ames, just over the hill in Mountain View. And other faculty here at UCSC, astronomers, are also funded by the NASA program. So the collaboration between UCSC and NASA has been quite beneficial to the campus and our involvement in the NASA space program. So without any further ado, I'd like to welcome our lecture tonight, and I think we'll find it quite entertaining. David?
Well, thank you very much for that nice introduction, and thank you all for coming out tonight. It's a pleasure to be here. I'm going to start by explaining that title that you've been reading up there. What was life? What is life? What will life be? Erwin Schrödinger wrote a book in just after the Second World War, and the title was, What is Life? Schrödinger was a very well-known physicist, a Nobelist, in fact, and he's one of the rare physicists at that time that took an interest in, actually molecular level and almost the atomic level. What is life all about? So that was the title of his book. Francis Crick cited that book as the co-discoverer of DNA, as one of his inspirations. However, since Schrödinger's time, we have learned a lot more about life on Earth, and we can begin to ask the question, what was life? Because life had a beginning. We're going to assume that it began on the Earth by the laws of chemistry and physics acting in an environment. We're also interested in what will life be, and what that means is that some of my colleagues, and we're trying this as well, are interested in putting together a form of artificial life. There's a name for this. It's called synthetic biology. And here, toward the end of my talk, I'm going to show you that's being made. We're getting to the point where we think that we're getting pieces of the life process put together in a cellular compartment. And I'm going to show you some images of that. Well, what about this word astrobiology? Let's see. Come on, let's go. There we go. That is a strange word, isn't it? Astrobiology. When this word first came out in about 1997, it was invented, in a sense, by the NASA administrator, Dan Golden, in collaboration with Wes Huntress, who is one of the people involved at the administrative level at NASA. And they made up this word to take account of the fact that this Mars meteorite, you remember that? Mars meteorite had been reported by NASA scientists to have fossils of microorganisms left over from perhaps life on Mars. That made a huge impression on the American public and on the world public, because for the first time there's a possibility of scientific evidence of life elsewhere. Well, NASA is nothing if not driven by curiosity science. In other words, it's the only funding agency that gives money to these sorts of research projects. So they invented a new field called astrobiology, and they put money into it. The first dollop of money was something like $10 million. It's given out in competition to astrobiologists like myself, who would put together teams and make a proposal for a research effort at their campus. Now, my biology colleagues sort of sniffed at this. They weren't very impressed by astrobiology, and they pointed out that if you took the bio out of astrobiology, it was a little bit more, what they thought the flavor was. However, we persevered, and I am happy to say that I am, in fact, on the editorial board of a journal that started up a few years after that name came along, Journal of Astrobiology. So this is real stuff, real scientists work. And scientists are attracted to new money like bees to honey, and we really have a lot of good people now working in 13 centers all over this country with some international connections in this new field of astrobiology. Well, one way to define astrobiology is it's the evolutionary narrative of how stars give rise to life. Now, isn't that interesting? We've thought of evolution as being just in the biosphere, right here on the Earth. And of course, Darwin, nearly 200 years ago, his birthday is going to be celebrated next year, by the way, so you're going to be hearing a lot more about Uncle Charlie as time goes on into next year. We really thought that evolution was something that happened on the Earth. But what astrobiology does is to try to take our imagination and real results as well out into space and look for the narrative, the story of how things happening in stars and in our solar system impinged on the early Earth to give rise to life. So that is one of the things that I'll be talking to you today about. Well, another way to think about astrobiology, however, is that it is the fact that hydrogen is a colorless, odorless gas that, when given enough time, changes into people. Yeah, well, you think that's funny, but it's the truth. And what I'm going to do tonight is to try to carry this story right from the beginning of time, beginning of our universe, on through to what we are here in the biosphere today. So that's really what I'm going to try to do for you. One of my boyhood heroes was George Gamow, and I didn't know how to pronounce his name, so he was George Gamow to me for many years until I learned a little bit better about pronunciation. He wrote a book called 1, 2, 3, Infinity, and I was a teenager. I read that book, and it was just great. I loved his ability to explain rather complex scientific results for people like myself, just a teenager. Well, George Gamow conceived that the universe might have a beginning. And he was one of the first two people to think along this line. And he really published this, and he made some predictions about the fact that if there is a beginning of the universe, you can predict certain things that are testing this idea. Fred Hoyle was a contemporary, and he did not think much of the idea of a big. of a beginning of a universe, in a radio show, he referred to it jokingly as the Big Bang, and the name stuck because Gamow's predictions pretty well came true, and the consensus now is that it is not a steady state universe that we live in where everything has just been the same infinitely back into the past and into the future, but in fact that our universe seems to have had a beginning. We call that the Big Bang. We can get a pretty good estimate of how long back in time this is. It's about 13.7 billion years is the latest estimate of this. And what you notice is that after the Big Bang, nothing happened for quite a while until about 400 million years later, when finally stars began to form. And the stars, of course, began to cluster into galaxies under the forces of gravity. And then through time, from that period to now, we have the universe that we live in. Our sun came into being right about here, about 5 billion years in the past. And we have a few billion years to go, so don't worry, we're still okay. But in fact, there is going to be an end to our sun, and you're going to hear about that in just a few more minutes. Now I want to talk about the elements of life, because we have to, part of understanding how life began is understanding where the elements came from and how the elements turned into molecules and how the molecules turned into living organisms. So these are the elements we call the biogenic elements, because they make about 99% of a typical living cell, of the mass of a living cell. Of course, hydrogen and oxygen are there as water, so that's a lot of mass. Carbon is there in virtually every organic biochemical that is parts of life. Nitrogen is very much part of amino acids. Sulfur is also in certain amino acids, and phosphorus is in nucleic acids. So these are the compounds, the elements that give rise to life. Where did they come from? It begins with hydrogen fusion. So this is the first step toward life. I told you that hydrogen, given enough time, turns into us. This is the first step. It's still going on. We know now that stars and our sun run on the energy of fusion that has to have temperatures about 10 million degrees to occur in a star when it begins to shine. And that hydrogen then turns into helium and releases a huge amount of energy. This is the most energetic physical process we know about. So here are 4 hydrogen nuclei, a very tiny little thing called a proton, and those 4 fuse at these temperatures into helium, and they release fusion energy. Just over the hill in Lawrence Livermore right now, scientists are trying to put together a simulation of this. There's going to be a whole bank of lasers focused on a little bead containing some isotopes of hydrogen. They hope that when they turn those lasers on, we might have a controlled fusion reaction that we can use as an energy source. Okay, here's the story. After millions to billions of years, depending on what kind of star it is, stars run out of hydrogen. They get much hotter, they shrink down, they condense, the temperature goes way up, 100 million degrees, and other fusion reactions begin to produce heavier elements, such as the biogenic elements that we're interested in. This is what our Earth and the atoms in your body, the atoms in the biosphere, that's where it comes from. Finally, a star undergoes an explosion which ends its life, and the elements are injected into the interstellar medium as dust and gas. Okay, now get ready, because here it comes. This is the Crab Nebula. About 1,000 years ago, Chinese astronomers recorded the original supernova explosion that now has resulted in what we see here as the Crab Nebula. This is several light years across. This is a composite of 3 different space telescopes. One that shows us the X-rays being given off by that original explosion. Another that shows us the sulfur and oxygen that is coming out into the interstellar medium around the original exploding stars. You can see that sort of reddish color. That's the dust that is giving off infrared light from the heat of that explosion. So here is one place where elements come from. All those elements that I showed you being synthesized in that big star that's gone through supernova have now been blown out into space and begun to diffuse through the rest of the galaxy. Now, what I want to show you here is something quite interesting. That little star, the remains, is still spinning. It spins about 30 times per second. You could hear a low hum. It's also oscillating. And these waves that you see coming out from that central star are a time-lapse movie. And those waves come out over a period of months, all shrunk down into a few seconds here. But it's really quite extraordinary. This is one of the rare times when a movie of a star can give us information about that star. Well, here are some of the people that were involved in working out the origin of the elements. Margaret, Jeffrey Burbidge, husband-wife team, William Fowler, and there's Fred Hoyle again. They co-authored a paper that came out in 1957 called Synthesis of the Elements in the Stars. And this paper, for the first time, put all of this together into one package that physicists could then believe. As a result, one of these people got a Nobel Prize. That was William Fowler down at Caltech. Fred Hoyle missed out on a Nobel Prize, even though he gave a very important part of this story, which I'll show you in just a minute. He was, however, knighted by the Queen, so he is Sir Fred Hoyle. The Burbidges are Emeriti faculty at UC San Diego and still pretty active in their fields. So this is Fred Hoyle's idea. He had this back in 1947, 46, and published it about that time. And he realized that if the temperature was high enough, that helium could begin to fuse and turn into beryllium. This is a metal nucleus. The beryllium could pick up another helium and fuse to form carbon. And there is that element. The carbon of your bodies and mine, the Earth, the other planets, really was produced in very high temperatures of these exploding stars and then blown out into space as the star broke up. The last part, the last of this little sort of scientific stuff I want to get across to you is where do the oxygen and nitrogen come from? Well, it's very interesting because at the same time as the stars go to high enough temperatures to form carbon, the carbon can pick up another proton to become one of the nitrogen isotopes. It can decay into another isotope of carbon called carbon-13. There's some of this in our bodies as well. Carbon-13 can pick up another proton, become a nitrogen isotope. Nitrogen can pick up another proton, become oxygen, and the cycle goes round and round and round, called the CON cycle, and this is where the oxygen and nitrogen of our biosphere come from. Made in stars, delivered to the Earth during accretion. That's part of the story. It's the first part that I want to get across to you. Well, it's really beautiful now to look out with the Hubble telescope. We see images that only our generation of human beings has ever seen, and only because we have these orbiting telescopes that give us a vision of what is really out there. Here are a couple of ordinary stars, like our sun, and they've reached the end of their lifetime, and they have also gone through an explosive ejection of these elements from their interior into outer space. By the way, these are sort of cosmic Rorschach tests, and the first astronomer who sees this gets to name them. So I don't know if you can see an Eskimo there, but whoever found this thinks that looks like an Eskimo. And the cat's eye, that sort of makes sense. Again, the blue is X-rays. The yellowish stuff is oxygen and sulfur, and then there's some dust around that stuff too. So isn't it interesting that stars go through their lifetime, then they explode, send the stuff out into space, and that then gives rise to the birth of new stars. So since the beginning of our universe, 13 billion years ago, there have been several generations of stars like our sun come and go, and many other generations of stars that started out too big, they explode, they go through a supernova explosion, and they also contributed to the rest of the universe. So here's not our galaxy, but this is what our galaxy would look like. This is called the Whirlpool Galaxy. And you're all familiar with the fact that this light is billions and billions of stars, as Carl Sagan used to say. 100 billion stars in our galaxy is one estimate. But what I want you to notice is this dark stuff here. That dark stuff is the ashes, stars that have gone through their lifetime and gone through this explosive release of their elements into the rest of the galaxy, and that is the birthplace of new stars and planets, as you'll see. So here is a sort of a close-up view of one of these. If we look deep into this with a telescope, we would see what we call a molecular cloud. It's a star-forming region. There's going to be a star starting up right there. That we have seen some stars now going through this process. Here is one such star. You can see this kind of disk of dust and gas that from which the star is drawing in the mass that is going to give rise to that star. You can also see the star beginning to shine on the dust around the edge here. Finally, out of that comes a solar system, and I'm going to show you how that happens next. This is what a dust particle looks like. I've been telling you about dust in our galaxy and the kind of dust that brings stuff to the Earth when the Earth was forming. This is about the size of a bacterial cell, micron dimensions. This might be 10 microns across is my guess. It's nothing but a silicate mineral. It's kind of glassy stuff. Silicon, oxygen, that's silicate. So it's a glassy mineral. And then on the surface of it, you can't really see it here, but on the surface there are accumulating layers of organic compounds. And that is a key to much of what I'm going to tell you now for the rest of my talk. And that is that organic compounds can be made in these intermolecular clouds, these interstellar molecular clouds, made by photochemical reaction of UV light, ultraviolet light, with the stuff that is accumulated on these dust particles. So some of the molecules that are delivered to the Earth, in fact, were made under these conditions. So let me go a little bit further here. This is really a beautiful image. This is a teacher's delight because it shows everything that I've been telling you about. It shows you molecular clouds. There's one of the clouds there that you can see, interstellar dust cloud. You can see another one up here, and these are being illuminated by the light of these new stars that have just formed recently in this particular region of our galaxy. This is all in our galaxy, of course. This is a nebula within our galaxy and not some faraway place. So these are new stars, and if you look up here, you'll actually see a star that has already gone through its life cycle. It has already exploded, and here's a ring of material coming out from that exploding star, and a couple of puffs of new ashes and dust that you can see coming out from either end of the star. That's probably the axis of rotation you see here. Now, the last thing I want to show you here, though, is this guy. Take a look at that. That is a solar system to be. It is a dusty star. You can sort of see the dust around here. And in the interior of that dust, lit by these other stars and probably pretty soon by the interior star there, is the dust that's going to turn into planets. So let's talk a little bit about that. This is an image out of a recent Scientific American article by Doug Lynn, who's one of the great astronomers we have here at UC Santa Cruz. And he's interested in planet formation. He's using computers to visualize what happens during planet formation. So in these sorts of computer simulations, what you do is to put into the computer's memory a whole set of particles, literally thousands of individual particles. Then you start them spinning, and then you let gravity interact between those particles. And what you discover is the particles begin to aggregate into these swirls you see here. The swirls get thicker and thicker and finally break up to form these planets that you see here. Now I'm going to show you, I think, the next slide. Let's see, I might have to, yeah, here we go. This is a little cartoon of what I've just shown you. You see a rotating cloud as one of those molecular clouds. Here's a solar nebula. This is a rotating disk. You're going to see the sun up here in the center. See how it's pulling stuff in? That's how stars get born. Now you're going to see planets begin to form from the extra dust around this star. The light of the star begins to blow away the dust particles around the sun, leaving behind the planets in orbit around that star. Now, this happens all over our galaxy. We now know of well over 100 stars that have planets surrounding them. We call these extrasolar planets. So this is a process that is ongoing in our near galactic neighborhood, and of course we think that something like this happened when the Earth was born. I'm going to show you another little movie picture, a cartoon, and what you're going to see is the Earth as it was very early in its history, maybe 1/100th of its present size. And you're going to see meteorite strikes hitting the Earth because this is how planets grow, at least terrestrial planets like ours. Their gravity picks up smaller particles from around them, smaller, we call them planetesimals. Those crash into the surface and produce the ever-growing planets. So you see there, you see these flashes. Notice this is turning a little bit. The artist has shown the turning, and each of those flashes is adding mass to that early version of the Earth. So we think that this is a pretty good understanding now of where the Earth that we live on came to be very early in its history. Well, something really amazing happened. Very shortly after the Earth grew to nearly its present size, another planet the size of Mars collided with our Earth. We're the only planet in the solar system that had this large an object crash into it. Just an accident that they happen to have intersecting orbits. This is now the consensus view of how the Earth-Moon system formed. And what I'm showing you are images. These are paintings done by Bill Hartman, who developed this idea. He's an artist scientist. So as he developed this idea, very respected astrophysicist, he would make paintings of these things. If you look him up in Google, which I just did, you'll see a whole collection of these magnificent paintings that he's made throughout his lifetime of astronomical subjects. Well, the heat generated by that collision was so great that the Earth melted. We are now standing on the Earth's surface looking out at the moon. The moon was only 1/10 as far away as it is now after this collision occurred. It's slowly moving away from the Earth. The moon grew out of the debris that formed a ring around the Earth for a few million years. And notice that red hot temperature. That's important to my story because these are temperatures so hot that organic material cannot survive. These are called pyrolytic temperatures. That means to break up with fire. And I've been to Hawaii and watched the lava come over plant life. You know, the plants just disappear. They turn into carbon dioxide as a result of that. And that's what happened on the early Earth. So the question is, where did all the organic molecules come from that were necessary to start up life on this Earth after this event? And the answer is, at least in part, that they were delivered to the Earth by the infall of comets, meteorites, dust particles, all of which brought organic carbon molecules to the Earth again. Now, some of you probably remember from your college years that we had another idea, though, that all the organic molecules were made at the Earth's surface. This is Stanley Miller's idea. Stanley just died a couple years ago. I knew him pretty well. He was down at UC San Diego, very respected scientist. And he and Harold Urey, during his graduate career, worked out a chemistry of molecular origins such that primitive atmospheres would turn into organic compounds. Well, the problem is that we now know that the early atmosphere was mostly carbon dioxide. And they thought it was ammonia and methane and hydrogen and water vapor, but it wasn't. It seems to have been carbon dioxide. So we were looking around for other ways to get organic carbon to the Earth, and this is what we've come up with. By the way, this idea has been around for a number of years. It's not my idea. Carl Sagan has been involved in this. Del Sim has been involved. John O'Rourke was the first to suggest this, that organic compounds were delivered by cometary infall. So here's another Hartmann painting showing a comet striking the moon and leaving craters that we still see today. You know, we're really confident about this story because we can look at the moon and see the craters that also would have been produced by impacts on the Earth. Here's a comet hitting the Earth. Here's another comet hitting the Earth. Here's that debris ring out of which the moon grew. And next time you look at the ocean, keep in mind that our best estimate is that it's about 10% comet water. So oceans are 10%. The mass of the ocean is comets, and comets are about 10% organic material. So as comets slammed into the Earth, we really think that they delivered quite a dose of organic compounds to the early Earth. These were then involved in life's beginning. So let me just summarize what I've told you at this part of my talk. Here are the dust particles of intermolecular clouds, interstellar clouds. There are ice mantles on these grains. The ice contains water, methanol, carbon monoxide, and ammonia. We know that from radio astronomy results. We also know from laboratory results that if ultraviolet light hits this mixture, it turns into things like amino acids, ketones, amides, quinones, and amphiphiles. Now that's a word that you might not have heard of, but just keep in mind it means soap-like molecules. These are the molecules that form membranes, and you're going to hear a lot more about that in just a moment. So these dust grains aggregated by accretion, that just means coming together, form larger structures, into comets and planetesimals containing those organic compounds that were produced here. These then impacted the early Earth, as I've just told you, and delivered this material to produce a dilute solution of organic compounds in the early oceans. So that's the story of organic compounds getting to the Earth. We think this is what the Earth looked like about 4 billion years ago, just a little bit before life began. There were lots of volcanic activity, land masses resembling today's Hawaii and Iceland and other sort of island volcanoes. I hear my mic keep going off a little bit. The moon was a lot closer still. The ocean was probably pretty acidic compared to today. It was salty, and the atmosphere was carbon dioxide. So keep this in mind because I'm going to take you with me on a trip to Kamchatka, Russia, just a little bit later. And we're going to talk about what happens to organic molecules when we go to an environment like we think the early Earth was. Okay, let's talk about what organic molecules might be required for cellular life to begin. It's not too hard. We have to have a source of monomers, like amino acids, to form polymers, like proteins. Amino acids do get together into proteins. We also have nucleobases. This is things like adenine, guanine, cytosine, thymine, uracil. These are the bases that go into DNA and RNA. They're part of that polymer. We also have carbohydrates made up of pentoses and hexoses. So these are all the monomers of life. Then you have to have molecules that produce cellular compartments because you cannot start cellular life without a boundary layer around it. And that's where a lot of our research comes in. So we have this word amphiphiles. An amphiphile just means something that likes two different kinds of things. So if you take a hydrocarbon chain like octane and gasoline, if you put a carboxyl group or a hydroxyl group on one end, a water-loving group, that's an amphiphile. Soap molecules are amphiphiles. The lipids of your membranes of our bodies, of our cells, are amphiphiles. Cholesterol is an amphipile. So all of these things have the potential to form boundary layers. So you're going to hear a little bit more about that. These are this is what they look like. These are the large and small molecules of life. Again, just sort of summarizing, hydrocarbons have two of the biogenic elements, just hydrogen and carbon. That's where they get their name. And they're part of lipid molecules in the body that form membranes and store energy as fat. Carbohydrates, we've added oxygen, and these are things like glucose that very much part of the energy metabolism of our bodies. Glucose turns into cellulose of plants. It turns into starch that plants use to store energy. Amino acids, we've added nitrogen and sulfur. We're now up to 5 of the biogenic elements. And glycine, of course, is an example of that simple amino acid that is very much part of proteins. There are 20 amino acids that are commonly found in proteins. Finally, we have the monomers of DNA and RNA. And you might not have heard this word before, but these are called nucleotides. And now we add phosphorus. And AMP, adenosine monophosphate, is an example of a nucleotide, and that's the A of the 4 bases in DNA, A, T, G, and C. Well, now we can ask, where do they come from? And I've given you the answer. I'm going to show you now how we know, how we can be confident of the answer. Because we think, as I mentioned, that one possible source is comets and meteorites. In 1969, in the skies over Murchison, Australia, a fireball like this just flashed into the sky. It was just like a very long lightning flash, in fact, lit up the landscape. People looked up and they saw this fireball moving across the sky, and then it exploded. Then a few minutes later, all over the little town of Murchison, Australia, 100 kilograms of rocks fell to the ground from this meteorite. So it's called the Murchison meteorite, and it is full of organic compounds. It's one that we study in my lab. Here's one of the stones that we have worked with. So you can still see the fusion crust, because even after it exploded, these things are coming in pretty fast through the atmosphere, so that melts the surface. On the other hand, the interior is insulated from all that heat, so it stays cold. The heat does not penetrate the interior. This is why this meteorite is interesting, because it has a lot of the chemicals in it that are part of the origin of life. So here we've broken it open. We discover that the insoluble polymer called kerogen, which is kind of a coal-like material, just a polymeric stuff, not really very interesting, about 1.5% of the total mass, and about a few percent of that is what we call soluble organics. These soluble organics you have heard of. They're carboxylic acids, like fatty acids of fat. They're amino acids. You can see that little slice of this pie diagram. They are amides, aliphatic and aromatic hydrocarbons are there, aldehydes, alcohols, amines, and even a little bit of adenine, one of the purines of DNA, has been found. So this is why we think that we're on the right track when we imagine that a lot of organic material was delivered to the early Earth, because it's still happening. We can go out, pick up these meteoritic fragments, and measure the stuff in the meteorite. So I'll just take you through a few images that will show you why I think this is important. This is another of these meteoritic stones. We've broken it open, by the way, and you can sort of see the interior. This is very primitive stuff. It's never gone through geological processing. So it's never seen a volcano. It's never been pressure cooked or anything like that, like other minerals. So it's very primitive. We also know that it's about 4.57 billion years old, because other people, not my lab, but other labs, have measured the uranium to lead isotope ratio in this meteorite and other meteorites. And the number they come up with from the decay of uranium into lead, which has a half-life about 4.5 billion years, is this number, 4.57. So we take that as the age of the solar system. The Earth is a little bit younger than that. If we look at the surface of that fractured meteorite with a microscope, we can actually see the fluorescent material. This is the organic deposits throughout the meteorite. And we can extract this organic material. This is our work. And we do what's called chromatography with it. We put a drop of the extract down here. We run chloroform as a solvent system in this direction on the plate. Then we turn it sideways and run hexane, ether in this direction. And that separates these organic molecules into visible fluorescent spots that you can see. Up here we have the polycyclic aromatic hydrocarbons. Down here we have various derivatives of those hydrocarbons, all fluorescent. And right here we have organic acids. Now, I've brought some of this to share with you. So I've got, this being California, of course, I'm going to share this with a wine glass. I'm going to pour a little of this material into the wine glass, just a few drops. I'm going to smell it and tell you what I smell. Wow, this is a very distinctive aroma. It's dusty, it's oily, kind of sour. It's all those things that we've extracted from the meteorite. So this is the stuff I now have circulating in my bloodstream, ancient organic compounds that were around before the Earth was born. Now, a couple of my colleagues in chemistry, Phil Cruz and Ted Holman, make wine, and they will tell you that 4.57 billion was a very good year because that's where we all come from. Now, you get to smell it too. So I brought a little closed container of this. And I want to warn you, I guess stay back here to stay in the light. I want to warn you that this is the stuff that's in tobacco smoke. It's polycyclic aromatic hydrocarbons. It's much of what you'll smell, along with a little bit of acid and oil. So if you don't like the smell of cigar butts, you're not going to like this smell. So don't smell it. So, take that little word of advice. But you can kind of look at it. It's a yellowish color because the polycyclics in it. And if you smell it, you'll smell a smell 4.57 billion years old. So why don't you smell it and see if you agree. What do you...
Smells like cigarettes.
Yeah, it really does. So this is polycyclic aromatic hydrocarbons, very common compound in the universe. And as I say, it's really fun to share this because it's rare. You are a very rare few people that have ever smelled this aroma of outer space. What we did was to extract that material, and we put it in the microscope and let it interact with water, just as it would on the early Earth. So this stuff is being delivered to the early Earth. It dissolves in water. It forms layers on the surface of the oceans and lakes of the time. And we found something very interesting. It forms membranous vesicles. These are real membranes. This is the organic acid portion of that chromatogram I showed you. They're real membranes. They can capture molecules like fluorescent dyes, so we can see that there really are an inside and an outside. And what we have proposed is that things like this were the membranous boundary compartment of the very first kinds of cellular life. So this is, I think, sort of a consensus now. People have agreed with that. They don't have any better ideas anyway. I want to show you just a couple more, one or two more chemistry slides. We think that this is what is making those membranes. These are amphiphiles called fatty acids. You would know these as soaps. And these fatty acids have the ability to assemble into membranous structures. And the reason they do that is that they form bilayers. So the membranes of every cell, there's 100 trillion cells in the human body. Every cell is composed of a lipid bilayer as its boundary membrane. So here's one layer, here's the other layer. This is all water, all these little red H2O groups on either side. And here I've superimposed a molecule called a phospholipid to sort of let you see this in 3 dimensions. This is a computer simulation. And if you could see this in real time on the computer screen, you'd see it doing things like this. It is a fluid. All of your membranes are fluid, and all of those tails are violently moving around, rotating, literally billions of times per second as they form this boundary layer of your membrane. Now you also notice one last thing here, and that is there's lots of water here and lots of water here, but almost no water here. I think right there is one little water molecule, just about right. Oil and water don't mix. This is the oily interior of all the membranes of all life on Earth. And this is as essential to cellular life as DNA and RNA and protein, because without that bilayer, life could not exist. What we've done then is to make simulations of those membranes I showed you from the meteorite, because we know that there are fatty acids in it and some fatty alcohols. This is called decanol. These have 10 carbons each. And look at these beautiful vesicles. These are real membranes that are formed by this very simple system of molecules. We can also capture big molecules like DNA in these structures. So here we stain the DNA that we've captured with a fluorescent dye, and you can sort of see these what we call protocells being produced. So isn't that interesting? It's very simple to make cellular compartments out of these molecules such as we see in the meteorite. And that is why we think that we're on the right track to seeing how life might have begun. Okay, I'm about halfway through, so if you're still with me, I'm still got a few more slides to go. I want to show you how we think that life might have begun. So all life is polymeric. So the first life must have used polymers of some sort. But what is a polymer? Well, you're going to hear this word again and again. Monomers are the sort of the pearls of life, and if you put a string through it, you have a polymer. So monomers are things like amino acids and nucleotides, there's that word again, and you string them together and you get polymers such as proteins and DNA. There must have been a way on the early Earth to make polymers by some easy, relatively easy chemical process. So what I want to do now is to take you through why this is important. It's important because polymers have a sequence of material in it. The monomers are sequences, and in those sequences you can contain information. So you've all heard that genes are in DNA, and in fact genes are sequences, and it's fun to think about what that means. This is a string of letters called a sentence. So that makes sense to you, doesn't it? Because they are in sequence. Those are the monomers. The letters are the monomers of those words and of that sentence. This is a string of chords called a melody. And if you are a musician, you'll recognize this great melody by Bizet's opera Carmen. This is a string of bases called DNA. So each of those bases has a certain code that is used in life. Now, what we have done as a teaching tool is to turn DNA into a melody so that you can feel the sense of sequence that comes out of DNA. So to summarize, DNA is a sequence of bases. A melody is a sequence of notes. They both contain information, either genetic or musical. Therefore, we can translate DNA into music. We're just going to say, let's make adenine A, that's one of the 4 bases. We'll make guanine G. We'll make cytosine C and thymine E. And here is the insulin gene. I'm the only person in the world that can whistle the insulin gene from memory. There's TTT, GTG, AAC, CAA, and those are the triplet codes in DNA for the amino acid phenylalanine, valine, asparagine, and glutamine. Notice the author of this beautiful bit of melody over here. And the B chain of insulin has about a little over 20 notes in it, so to speak. So when I play this for you, you're going to hear these notes. So just listen now for this melody. You'll hear it just right at the start when I play this. Bum, 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 bum. So there's the first 4 amino acids and the first 12 notes. Okay, here we go. Listen for it. Uh-oh, we have lost the music. Where's my technical help? What happened? It's really fun to listen to this. I hope we can get it to go. There we go. Sorry. We'll play it again for you. I'm going to go back one. There we go. Okay, here's the melody again as music, and here you should hear it. This is the next chain, the A chain. Unless you are a severe, oh yeah. Unless you're a severe diabetic, the islet cells in your pancreas right now, islets of Langerhans, are making insulin. And they're making it at about the rate that you heard that melody, about 1 amino acid per second being added using the ribosomes in the cells of your islets of Langerhans. So it's really nice to actually see the rate and hear the music. So here's the deal. Origin of life occurred when polymers containing specific sequences happened to self-assemble in the prebiotic environment. So what we're going to ask now is whether that could really happen. We're going to go visit a volcano. I want to recall a little saying from Charles Darwin to you. Charles Darwin didn't really think much about the origin of life, but he speculated about it in a letter to his friend Joseph Hooker, 1871. But if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes. At the present day, such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed. So here's this great mind thinking ahead in an almost prescient manner about how life might have come to be. He knew about proteins, but he didn't know about amino acids. He knew that probably nitrogen was there, and he knew that phosphate was there. But notice he doesn't have carbon there. So he had really no knowledge of the chemistry of life that we do today. Well, we're going to honor Darwin. This is the 10-pound note of the Bank of England. And of course, he was English. And here's Uncle Charlie, as we call him fondly. Here's the beagle on the 10-pound note. And here's one of his ideas where a hummingbird co-evolves with a flower of a plant, one of the things they brought out in this 10-pound note. Well, I told you that the earth was warm, and in fact even hot at the time of life's beginning. It was volcanic. There were oceans, without doubt. There was a carbon dioxide atmosphere, and there was no warm little ponds, but there were hot little puddles. And in this image from the Smithsonian basement, we see a boiling puddle. And what we're going to do on today's Earth is to go exploring and see if we can find an analog environment. We went to Kamchatka twice now to find these places. The reason we went to such a faraway place is because recent volcanism and high altitude and high latitude produce sterile sites for the analysis we want to do. And we want to find if there's any organic synthesis that can occur under these conditions. This is what Kamchatka looks like if you were going over it in a shuttle, a space shuttle. We're 100 miles high or so. Japan is down here below. Alaska is several thousand miles up here. And to get there, you fly up to Anchorage, then you take over early morning flight, and you land in Petropavlovsk, which is right here, a former military base of the Russians, but they've now opened it up to tourist use. Then we hop in a troop carrier, and I'll show you that in just a minute. We take about a 4 or 5 hour trip to Mount Matnowski, which is one of the more accessible volcanoes. So here's the little gang that we took along. I don't think you know any of these people, but this is Jamie and Megan. They were graduate students over at Stanford when we took this picture. This is Jan and Jake. They were postdoctoral people at the Carnegie Institution in Washington. This is Vladimir Kompanechenko. He was the guy that suggested this trip and organized it locally. This is Igor, believe it or not, Igor. He was our local guide. And this is my buddy Tony Hoffman, who lives down the street and just came along for the ride. Here we are hiking up the volcano. This is an ice field. It's about a 3 or 4 hour hike to get up from where we camped, basically, up into the volcanic crater. And here's Vladimir looking down into the volcano. And boy, when you see something like this, it's very, very primeval. There's nothing alive here. There's no plants. We couldn't even find bacteria up. We don't know quite why they should have been there, but we haven't been able to see any. There's water, of course, rocks, and heat, and that is what the early Earth was all about. So here I am now, it's about 5 years ago, doing this work. I'm very carefully reaching down with a long pole into this fumarole, it's called, trying to get a sample of boiling material down here. We're going to analyze that for organic compounds. Sometimes we had to wear gas masks here, by the way. There were fumes of hydrochloric acid and sulfur dioxide. This is what we then pulled out. It's a flask full of mud and water. So we would then take that, store it, carry it home back to the lab, and look to see what was in it. Well, total surprise. There was nothing. We could find no organic compounds in this water, and we really expected to find something there. So we decided to go back again. But this time we reorganized Darwin's little conjecture. But if, and whoa, what a big if, we could conceive in some hot little puddle, not a warm little pond, with all sorts of prebiotic amino acids, nucleobases, lipids for membranes, phosphoric salts, light, heat, etc. present. So we're going to try that in Kamchatka. So here's the boiling puddle we saw, the hot little puddle. That's going to be our laboratory on Mount Midnowski again. By the way, you can see where we scraped up some clay to analyze the clay of this particular puddle. And the experiment was just very simple. We made what is called a prebiotic soup, a soup that contained amino acids for them, nucleobases, this is for ribonucleotides, phosphate, and myristic acid. So those are examples of all the kinds of stuff that we think might have been involved in the origin of life. So we wanted to ask, how fast do these degrade, and does any synthetic chemistry occur? Well, I tell, when I give a talk like this to academic audiences that have young faculty in it, I say, don't try this experiment until you get tenure. Because here I am, hopefully pouring my prebiotic soup into this puddle, wondering what's going to happen. And you can just decide for yourself on the wisdom of that experiment, but we found some very interesting things that guided us then. So what we discovered right away is that the myristic acid, that fatty acid, the amphiphile that we added, moved from the center to the edge. You can see this white stuff all around the edge. And so the idea that was given to me by that is, and with apologies to Michelangelo, that life began there. So this is my current idea that I'm trying out on you folks. This is what I'm putting in my book. And that is at the edge, not in the pool, but at the edge of the pool where you have dry, wet cycles. That is a place where you can pump organic chemistry uphill toward more complex materials without doing very much work on it. Very little chemical work. It's just driven by the fact that it gets dry and then it gets wet again. So that is what we're going to try to model in the laboratory. And I'm going to show you just a couple of results, just one result. This is the only technical slide I'm going to show you, and it's very easy to understand. I won't go through it in any detail, but every place that you see black stuff, that is a radioactive RNA that has been synthesized in the laboratory under the simulated conditions of that pool. So we really think we're onto something. Look at this. You just see all that black stuff there is RNA that's been synthesized under various conditions. So it really does work. We can make a long molecule of one of the primary molecules of life just by putting it into this simulated environment and pumping the system by cycles, wet-dry cycles. Okay, well, let me just finish up here. This is what I think happens. We have a delivery of organic materials during late accretion, giving these amphiphilic molecules that form membranes that I showed you from the meteorite. We also have geochemical synthesis of long molecules that sort of make a random mix. These long molecules then become encapsulated in these membranous vesicles to produce what we call protocells. So that is the start toward the origin of life, the first step. Do I have another 5 minutes, do you think, Todd, to finish this up? Okay, let me just hang with me a little bit longer. I'm going to show you how we and others are trying to make artificial cells, and then we're going to finish up. So let's start with some ribosomes that these are the protein synthesis structures in life today. So we can isolate these very easily. Let's put a membrane around them. We can do that. I've shown you how easy it is to put a membrane around big molecules, even things like ribosomes. That gives an inside and an outside and protects the ribosomes. They're in an environment they can't get away. Let's throw in some DNA. The DNA has genes built into it. The genes tell the cell how to make enzymes and how to make ribosomes. So this is maybe 200 genes that we would have in that little piece of DNA. That's how much it would take to make a minimal form of life. Let's make the ribosomes active. When they're active, there's 2 of them that stick together. We've captured an enzyme that can transcribe the gene from the DNA into what's called messenger RNA. That's going to carry the message to the ribosomes. You'll see how that works in a minute. So we've captured that enzyme, and we're going to add some amino acids, 20 amino acids, and we're going to put them in as what we call transfer RNA complexes, because otherwise nothing would happen. We have to activate them. We're going to add some nucleoside triphosphates, NTPs, and that would be AUCG of RNA. We're going to add deoxynucleotides, such as ATP and so forth, that will be the 4 bases of DNA. And then we're going to wait. Nothing happens. The reason is that this is outside and all of this is inside. So we have to think about how to make it permeable. So let's make permeases. We're going to stick some proteins into the membrane to make little channels. And this then will finally allow stuff to get in. At that point, everything starts. The RNA polymerase begins to make RNA. That's that little red thing you see coming off there. The RNA goes off to the ribosomes, begins to be read by the ribosomes. The ribosomes begin to make protein. There you have that. And the cell begins to grow. At least the stuff inside begins to grow. We can't leave the DNA behind, so we have to add a DNA polymerase. And the DNA polymerase, using the deoxynucleotides, can make more DNA. You can see that we're sort of doubling up the amount of DNA. We can't leave the membrane behind because that's got to grow, so let's add an enzyme that'll make more membrane. And we can do every one of those steps. In my laboratory, we would be able to do this. Is it alive? No. And the reason is that we don't know how to make the ribosome. We can have the genes up here, but getting those genes to systematically produce the proteins and the RNA for ribosomes, nobody's done that yet. Very complex sort of thing to try to do. Maybe it's possible, but not yet. The other thing is that there's no control here. All of these things are going on without any regulatory feedback mechanism. So this thing will just kind of grow and flop all over the place. So it's not alive yet, but it's a pretty good step toward life. Here is a paper that did pretty much just what I told you. It captured some ribosomes in vesicles. You can see those little black dots in these vesicles here. In fact, we made these just to prove it could be done. So these are vesicles with the ribosomes. They added a messenger. They added amino acids and ATP inside this vesicle, and he got a little bit of green fluorescent protein, which is the fluorescent marker to show that they were making protein. They very cleverly made a channel protein called alpha hemolysin. This then went to the membrane, put a hole in the membrane, and let more amino acids and ATP in. And these little guys went for about 4 days. At the end of that time, they were filled with a glowing protein called green fluorescent protein that is used in these kinds of experiments. So that was pretty impressive. That made a lot of news when it happened. So the origin of life must have even been simpler. You can't start with something like this. You can't start with ribosomes. You've got to start with a much simpler scaffold of life, maybe catalytic RNA. In fact, some people have suggested that we actually went through an RNA world, so that the chicken and the egg didn't come one after the other. They came together because RNA can make both catalytic functions and can carry genetic information. So some of my colleagues are trying to make this work in their laboratories. This is what it's going to look like. This is what the RNA world would look like. This is us. This is our work. We've put an enzyme in these lipid vesicles that can make RNA and got it to work. And you can just see the RNA glowing because we've stained it with a dye that makes it fluorescent. So I think this beginning of life might look like just sort of cellular structures of different sizes, all competing for nutrients and energy. And at some point then, one of these guys came alive. So in my talk, hydrogen to people, I've shown you how we can get up to protocells. The protocells turn into bacteria, first forms of real life for bacteria. And the bacteria slowly got their act together and over several billion years came up to life today. And this is my daughter, Austa, and my daughter, Stella. And I truly believe that they are made of stardust. Thank you very much.
Thank you, David. We'll have questions now, and there's a portable mic. Please hold up your hand, and we'll bring the mic to you.
Somebody's got to be brave. There we go.
I wanted to ask about organic molecules on the moon. It seems like we would be finding a lot of, or we would have found a lot of interesting organic molecules, or maybe some organic molecules on the moon, and some, and I don't, it could be my lack of knowledge about the issue, but I didn't really think that there was much found.
Yeah, good question. So the question has to do, are there organic molecules on the moon? The answer is no. There have been, at parts per million, we've analyzed lunar material, we being the scientific community, not me, and it's just not there. We think we know why. The moon has no atmosphere. Everything that hits comes in at 20 kilometers per second, really, really fast. It just vaporizes everything. Furthermore, the moon has very low gravity, so anything that vaporizes just evaporates, it goes away. We might find a little residual stuff if we could dig down deep into the lunar crust, away from all this pounding that the moon has gotten over its lifetime. There are organic molecules on Mars. We have Martian meteorites. We've probably a dozen or so. And when we look into those, we can find polycyclic hydrocarbons. That work was done over at Stanford. So there is organic material on Mars, not much, at least in these little surface areas that we've been able to scratch away at. I kind of expect that we're going to see a lot more when we get deeper into the Martian terrain.
Thanks. Hey, Dave. Are you excited by the prospect that any of what you're describing might possibly be happening now on Titan? And might there be a possible mission that NASA could conceive of to find out whether it is indeed happening there?
Yeah, Titan is one of these moons around one of our giant planets. Jupiter and Saturn both have big moons. Every one of them seems to be different, geologically and in terms of composition. And Titan is one that we've visited. I'll remind everybody that we've had a spacecraft deliver a little module to the atmosphere it's gone. We've actually had visual images brought back from Titan. Titan is cold. It's icy. The only liquid there is liquid methane and liquid ethane that might actually form seas on Titan. And the rocks there are not really rocks, they're just solid ice. So in my opinion, life would have a lot of trouble getting its act together in a place that cold, even given a fluid to work with. Of course, that's just my best guess, and I hope that I'm wrong. I'd love to be proven wrong. We think of Titan as a prototype, really primitive Earth, well before Earth came to be as a planet, and that the chemistry that goes on there, we certainly know that there's photochemistry in the upper atmosphere. It's got a slight yellowish orange color, in fact, from this. And so we think that some of the chemistry would be very interesting, but probably not leading to life. Somebody in the back? Yeah, you'll have to wait for the mic. It really helps. Otherwise, I won't hear.
Where did organic compounds come from in the first place? How did they get on the comets and meteorites?
Oh, okay. Question on how did the organic compounds get to the comet? Remember I told you about the dust particles. Those have that layer of ice. The ice is acted upon by ultraviolet light that makes photochemistry occur, and you get complex organic molecules piling up on those dust particles. Dust particles aggregate. The ice turns into cometary ice, for example, or meteoritic ice. And so all that organic stuff comes along when the dust particles bring it into the comet or meteorite, or asteroid, in fact, from which meteorites come. So that's where it gets there in the first place. I might add that we have radio telescopes. We can point into these molecular clouds, and we can actually see spectra from the organic material that's accumulated in the clouds. So it's there.
Thank you. You got us up to a, is this too loud? Am I talking too loud? You got us to a spot where we had a membrane filled with self-replicators. How does that membrane, does that item then self-replicate itself?
Yeah, okay, good question, and one that we don't have an answer to yet, but I can give you thoughts. First off, it's not just one of these little things. In a little test tube containing about 1 milliliter of water with 1 milligram of lipid in it, there are 1 trillion of these little vesicles. That's just in 1 milliliter. Now imagine a global ocean. Imagine, trillion? I guess not trillions of tons. Lots and lots of organic material. It's 10 to the 18th kilograms is one estimate of what was available on the early Earth. So imagine that sort of being cycled, going through these wet, dry cycles, being stirred and so forth, electrical energy operating on it, UV light operating on it, all of this stirring going on. These are all these experiments. We call that a global scale experiment in combinatorial chemistry. So we don't know which one of those is going to be the one that sort of catches fire and begins to be able to grow. If one does, here's what we do know. It's going to be polymers. It's going to have a source of energy. There's only a few kinds of energy that would be available to it. It's going to use ordinary molecules, amino acids, maybe nucleotides. We don't know quite how they could be made yet. And these polymers are going to start to talk to each other. And when you get that happening, then you close the cycle between information and catalysis, which drives all of life today. That's what it's all about. So that's what I'm trying to figure out, is to answer your question. How did that little compartment happen to catch just the right kind of stuff to begin to go through the process toward a living state? So that's about as good as I can do.
I know there are a lot more questions, but I'd like people to be able to enjoy the refreshments, and so why don't you join me in giving a round of applause to Professor Deamer.